Hey, it's Mr. Shrum, and welcome back to the Inheritance Patterns and Genetic Research tutorial walkthrough. We left off right before we started the Patterns of Inheritance section. So here we go. When Gregor Mendel was studying the traits of several generations of pea plants, he was observing simple patterns of inheritance. Each parent plant contributed one gene for a particular trait, and the combination of these genes allowed for the phenotype of the organism. In humans, a few traits seem to follow these simple patterns, but many traits have more complex patterns of inheritance. So you can see here, we have one parent and another parent, their first generation, and then their second generation. Recessive and dominant genetic disorders. Alleles can be dominant or recessive. And what that means is if one of the two alleles coding for a trait is dominant, then the dominant trait will be expressed in the organism. It dominates the other trait. And if a recessive allele show their effect when both copies of the allele are present in the individual. And I think we'll get a few more pictures in the next few slides to help you understand what that means exactly. But genetic disorders are caused by mutations in the genetic code. Many genetic disorders are hereditary in nature. Some of these genetic disorders are difficult to recognize because the defect may be present in the developing fetus, but the symptoms of the disease may manifest at a later stage. For example, it is not completely known why many elderly people develop Alzheimer's disease, but a few cases of Alzheimer's are known to be due to a mutated gene. People with these mutations are more likely to develop Alzheimer's before the age of 65. So here's a chromosome with alleles, and here's a mutated allele. See how this one's a little darker than this green allele? So that's a mutated allele. And if you're unfamiliar with Alzheimer's, Alzheimer's is a degenerative brain disease that affects neurons. Neurons are the cells in your brain and central nervous system. The disease leads to memory loss and the loss of other brain functions. The disease causes about 60% to 80% of cases of dementia, which is gradual memory loss. Very sad. And several methods can help researchers predict the occurrence of genetic disorders or trace the lineage of an inherited genetic trait over the course of generations. We have pedigree analysis. That is a method of studying the genetic family history of a particular individual using pedigree charts. And these charts use different symbols and shapes to show traits in existing generations. Pedigree analysis also helps predict the possibility of genetic disorders being transferred to individuals. The charts can be used to predict the genotype and phenotype of future generations in terms of the children being homozygous or heterozygous for different traits. The charts help determine relationships between dominant and recessive traits in individuals. And I'm going to click on the genotype definition. That is an organism's entire genetic code, including genes that are not expressed in the phenotype. So think of genotype, gene, genetic code. That's their entire genetic code, while We saw another definition earlier, phenotype. That is the expressed traits of a given organism. So I think of physical, if you can physically see the traits, those are the phenotype. Genotype is the genes that may or may not be expressed. So the entire genetic code is the genotype. The phenotype is the expressed traits. Punnett squares. This is another method of analyzing genetic traits. And these squares can be used to determine the probability of a particular trait being inherited by the offspring on the basis of parental genes. So let's look at how some traits cause disorders in humans. Recessive genetic disorders. In recessive gene disorders, both mutated alleles from one from each parent are present in the individual. If a single recessive allele is present in the individual, the person will be a carrier of the mutated allele for that disease. This pattern is true for most chromosomes, except for recessive genes or X chromosomes. Females inherit 2 X chromosomes, and males inherit 1 X and 1 Y chromosome. Therefore, males need only 1 recessive allele on the X chromosome to develop the disease. Genes on the X and Y chromosomes are called sex-linked traits, which we'll discuss later in this lesson. For now, let's look at diseases on non-sex chromosomes that are caused by recessive alleles. Cystic fibrosis. Maybe you've seen this in commercials, but cystic fibrosis is a condition caused by a mutation in a gene on chromosome 7. The mutation affects mucus-producing glands, digestive enzymes, and sweat glands. The disease results in the formation of thick secretions, and the secretions accumulate inside the organs and glands, blocking their normal function. An affected individual will have both mutated copies of the allele. Tay-Sachs is a fatal disorder that appears in infants around 6 months of age. It is caused by a mutation in a gene encoding for an enzyme. The enzyme responsible for breaking down fatty acids is absent in this disease. The lack of this enzyme leads to the accumulation of fatty substances in the nerve cells. And that leads to the degeneration of neurons. The mutation affects the gene present on chromosome 15, which codes for the production of the enzyme. Affected individuals will have 2 copies of the mutated gene in their body. So here's a picture of 2 parents. Father is a carrier of, well, in this example, it's cystic fibrosis, and the mother is also a carrier of cystic fibrosis genes. So their children, there is a 25% chance that this child will have cystic fibrosis. So if they had 4 children, let's say, one child would be healthy because they received both normal genes from the parents. Child 1 will be a carrier, but still not cystic fibrosis affected. And so will this child. However, 1 and 4 will receive both recessive mutated alleles, and thus they will have cystic fibrosis. Albinism. Albinism is a disorder characterized by the lack of an important pigment in the skin, hair, and eyes. Special types of cells called melanocytes produce a pigment called melanin that imparts color to the skin and eyes. Genetic mutations occur in genes responsible for enzymes that help in the production of melanin. Such mutations mean either less melanin or a lack of melanin, leading to albinism. Humans with albinism can have a range of eye and skin color, depending on the amount of melanin in the skin and eyes. However, albinism in humans always leads to poor eyesight because the colored portion of the eye is somewhat translucent. Individuals with albinism will have 2 copies of the mutated gene in their cell. So those were recessive genetic disorders where you needed both mutated alleles from both of your parents. Both of your parents had to be carriers. Now we're going to look at dominant genetic disorders. And this is caused by the presence of the gene on just one of the 2 inherited chromosomes. So let's explore 2 of these dominant disorders. Huntington's disease is a progressive brain disorder that leads to uncontrolled movements, emotional problems, and loss of brain function. Individuals with the disease begin to experience symptoms in their 30s or 40s. The disease is caused by a mutation in a gene that is believed to play important roles in nerve cell functions. Affected individuals inherit the dominant gene with the mutation. Achondroplasia is a form of dwarfism. People with this condition develop a short stature because cartilage fails to develop into the bones. And the disease affects the gene that provides instructions to make a protein for the development and maintenance of bone and brain tissue. It's an autosomal dominant disorder. The affected individual has the mutant dominant allele. So here's an example of the Huntington's disease. Only one parent had to be affected. And the chance of their offspring getting it, 50% normal, 50% will receive this dominant gene. So it's a higher percentage than recessive genes. Galactosemia is an inherited genetic condition. Children with this condition cannot break down the sugar galactose, which is part of lactose. Based on the pedigree chart, is this condition a dominant or a recessive disorder? Explain. So we're told that both parents are unaffected, but they are carriers. One child is unaffected. 2 are carriers, but unaffected. And then 1 is affected. So there's a 1 in 4 chance of being affected by galactosemia. So that means it's recessive. Both parents are heterozygous for the trait, but they do not exhibit the disease themselves. If it were a dominant gene, both parents would have the disease if they carried the gene for it. That's another quick way. So if you see carriers, that means it's a recessive. If it's dominant and you have it, you can't be a carrier because you actually do have the gene, the disease. For children who do not have galactosemia, what are their possible genotypes for this trait? So there's heterozygous and then homozygous dominant. So this means one gets um, is all the dominant traits, and then some are a mix, like carriers. The children may either be carriers, making that heterozygous, and they will not have any recessive genes. So think of homozygous dominant as you don't have any of the recessive genes. This guy didn't get any of the recessive genes, so he's not a carrier and he's not affected. He's homozygous dominant. Carriers are heterozygous. Complex patterns of inheritance. So far, we've explored traits controlled by two alleles, either dominant or recessive. However, many traits are not simply dominant or recessive. They have complex patterns of inheritance. Let's look at some of them. Incomplete dominance. We've seen that for a dominant trait to be expressed, the dominant gene needs to be present in the cell. In incomplete dominance, there are two alleles. One is dominant and the other is recessive. If the dominant allele does not completely mask the effects of the recessive allele, the observed phenotype can show a blending of both alleles. So, for example, in snapdragon plants, a cross between homozygous dominant red flowers and homozygous recessive white flowers produces heterozygous pink flowers. Makes sense? Red plus white equals pink. The heterozygous result does not have the same phenotype as the homozygous dominant form with both the dominant alleles. Instead, it's a blend of the two phenotypes, dominant and recessive. So you can see in this square how we determine that. We have R for red and R, and then we have R prime. We'll say that means not red. And the white has both not red alleles, and they have both red. And then in the square, you match this letter with this letter, this letter with this letter, this letter with this letter, this letter with this letter. So you see, they're all a mix of red and not red, red, not red, red, not red, red, not red. Codominance. Codominance is a form of inheritance in which both alleles of a gene pair in a heterozygote are fully expressed. As a result, the phenotype of the offspring is a combination of the phenotypes of the parents. It's called codominance because the alleles are equally dominant and expressed, not blended together as with incomplete dominance. An example of codominance is the speckled hen. In some cases, when a homozygous black hen is crossed with homozygous white chicken, the result is a heterozygous speckled chicken with a mix of white and black feathers. The speckled chicken inherits alleles for both white and black feathers from its parents. So we have black chicken with two gene pairs of black, white chicken with two gene pairs of white. All right, so sickle cell anemia is a disease that causes the blood cells to be shaped like sickles instead of being the normal circular shape. The table describes the phenotype of red blood cells when a particular genotype is present. What type of inheritance is shown by the genes that control sickle cell anemia? Explain your response. So we have homozygous recessive sickle cells, homozygous dominant normal circular blood cells, heterozygous, subnormal cells, and some sickle cells in the blood. This resembles a codominant pattern of inheritance because the heterozygous forms show both traits equally. So if you do have some normal cells and some sickle cells, that's codominant. Now, if they were just a carrier, that would be a different story. That would just be a recessive gene. But this is codominance. Multiple alleles. Some traits are controlled by many alleles of a particular gene. An example is human blood type. Blood type determines the kind of blood a body will accept or reject. During a blood transfusion, the blood type of the recipient and the donor must be compatible. Otherwise, the recipient's body will just reject the donor's blood. And there are 4 blood groups: A, B, AB, and O. Blood type is determined by the type of antigen on the red blood cell. And this antigen is any substance that triggers your immune system to produce antibodies. So antigen, antibodies. A and B are 2 types of antigens that can be present on the red blood cell. The allele for blood type A is IA, and the allele for type B is IB. When people have neither A nor B antigens in their blood, they are in the O group. This blood group is denoted by the recessive allele I. So here's human blood types and their genotype. So phenotype A, they either have 2 IA or 1 IA and a recessive I. B has 2 IBs or 1 IB and the recessive I. AB has a dominant IA and a dominant IB, and O finally has 2 recessive Is. All right, oh, whoops, it already answered, but based on the genotype of AB blood, what relationship do the A and B genes have? They're co-dominant. Neither of them are recessive. The A and B antigens are equally present on the red blood cells, so the relationship of the A and B genes is co-dominant. When you donate blood, it can be used for blood transfusions. Transfusions are common during certain surgeries or when a person has an injury that causes a lot of blood loss. The O blood type is considered to be a universal donor because a person with any blood type can receive it. However, people with the O blood type can receive blood only from those with the same blood type.